Samantha Machado.
And my name is Grace Tirado.
And we are second year bioengineering undergraduate students at UC Riverside.
And we are currently doing research in Dr. Joshua Morgan's Tissue Injury and Mortality Engineering Lab within the Department of Bioengineering.
And currently we are working with colon cancer cells called HT-29, and let's go into more depth with it.
Hey everyone! So what exactly are HT-29 cells? HT-29s are cancer cells that are typically found within the colon. When a certain drug, known as MTX, is added to the cells, they may secrete mucus. The type of mucus that is produced corresponds to a specific segment of the gut. Our aim is to gather this gut data in order to create a tissue culture model in order to better understand gut and mucus interactions. More specifically, we want to study how bacteria binds to mucus within the gut, as well as its possible effects. Here's a demonstration of how we care for and treat the HT-29 cells for our experiment.
For 3 months now, we have been working on this project of treating HT-29 cells. For this experiment, we have the cancer cells seated in 90mm tissue culture plates. We use this type of plate because tissue culture plates provide cell adhesion. In other words, they allow the cell to grow on the plate. Right now, I am adding new media, which is the liquid you see that I am pipetting from a tube. Media is like food for the cells. It has the nutrients they need to grow and stay alive. There are different types of media used for each cell line. So for this one, we use one that's just for the HT-29 cells. I am also adding MTX to the cells. MTX, which is short for methotrexate, is a chemotherapy drug that stops the cancer cells from growing. For those who don't know or don't fully understand what chemotherapy is, let me explain in a simpler way. Chemotherapy is a treatment that uses anti-cancer drugs to kill the cancer cells. In our project, we are dosing the cells at a low concentration, since in our case, it encourages growth of the HT-29 that secrete mucus. So far, we have just been observing how MTX affects the colon cancer cells in order to obtain data regarding their mucus expression.
This is a visual representation of what our cells look like. When we first start to culture them, they look like this: small, individual, and circular. Once they are fed and allowed to grow, they form these larger colonies. This is an example of the differences in behavior that we are studying.
In terms of application, we hope to use our data and model in order to better understand a possible gut-related disease, obesity, as well as its origins. There's a theory that obesity may stem from gut bacteria traveling from the mucus layer into fatty tissue regions. A gut model may help illustrate interactions like these. As you can see, studying HT-29 cells is beneficial since it could better our understanding of the gut processes in which may help us against the struggle against obesity.
I hope you all enjoyed this video, and I want to take a minute to thank our wonderful PI, Dr. Morgan, as well as these amazing sponsors for supporting us.